Hello, class. So this is the second part of the series Biosynthesis of Amino Acids. And so what are we talking about in this second part of the series is the amino acids are made from intermediates of the citric acid cycle and other major pathways. So we'll start looking at this diagram to kind of describe the biosynthetic families of amino acids. And what it has is the major metabolic precursors are shaded in blue, listed here. You got the oxaloacetate, pyruvate, and all these other metabolic precursors that ends up making amino acids. And so the amino acids that give rise to other amino acids are highlighted in the yellow right here, as you can see. And then you'll also notate that as far as essential amino acids for humans are bolded and are all bolded. And so, as far as making aspartate, aniline, and glutamate, that is formed by addition of an amino group to an alpha keto acid. And so, what happens is right here you have this internal aldamine, and why it's internal is because it is attached to an enzyme site where the residue is lysine to directly attach to this key intermediate, the aldamide. And so then you have glutamate that releases this bond from this internal enzyme to end up having alpha ketoglutarate form, and you have this free amino group that is here. This is called the PMP structure, and then from there you have different derivatives of an alpha keto acid that is bound to this free amino group, and then that forms this ketamine, and then you have deprotonation to form that quinonoid intermediate. Then you have protonation to form this now external aldamine, because you have this aldamide structure that you can see matches here, but instead of being linked to an internal enzyme, it is linked externally, not to any sort of internal enzyme. And that is ultimately released to have your amino acid, and then the aldamide is back to being bound to an internal enzyme. And so just try to be familiar with some of these steps, the internal aldamide to PMP to ketamine to quinonoid intermediate, and then external aldamide, and then back to the internal aldamide. And then from here, you have the formation of asparagine from aspartate, which is listed here. So the first step is an ATP dependent type of reaction where you have a pyrophosphate group is released, and what is attached is the adenolate that's attached to there, and then you have some free ammonium ion that replaces the adenolate, and then you have a free amino group here, which this process is more so like a transfer amination, and you finally have that form of the asparagine. And then as far as glutamate, that is a precursor for glutamine and proline and arginine. So we already previously discussed glutamate into glutamine, but this slide talks about glutamate to proline and arginine. And so what you have is, starting with glutamate, you have a ATP, where it forms an acyl phosphate right here. So a phosphoryl group is attached here, and then it gets replaced by the NADPH, and that's where you have that hydrogen that is there. So whenever you see NADPH, that is reduction. And then forms this intermediate that can go in two directions. Either go through dehydration to form a cyclic structure, and then you have NADPH again. So remember that's reduction. So adding the hydrogen right here, and then you finally have that form of proline as far as amino acids. And then the other direction is a reaction with glutamate to form alpha ketoglutarate, and also the ornithine. And remember, ornithine should look kind of familiar because we've seen this before in the urea cycle. And then from there, it goes through several other steps to end up forming the arginine amino acids. And then as far as another precursor that we see in glycolysis is the 3-phosphoglycerate, which is listed right here. That is used to make different amino acids. For this one is serine, where you have this NAD+. So when you see NAD+, that's oxidation. And so that oxidation is happening right here. When you form that double bond, then it reacts with glutamate to transfer over the free amino group, so that transamination, and you have free alpha ketoglutarate. Then you have this intermediate, and then from there you have hydrolysis formed to get rid of the phosphoryl group there and replace it with the hydroxyl group, and you have the final form of the amino acid. And this particular amino acid can react with tetrahydrofolate to end up forming glycine, excuse me. And then it also formed this methylene tetrafolate, which reacts with homocysteine to end up forming the methionine. And so homocysteine, that comes from actually SAM. So remember this methyl donator, and so that's why that's highlighted here. It donates a methyl group to a molecule, and then from there you have this S-adenosyl homocysteine, where you can see you can have some cleavage that happens right here to end up forming the homocysteine, and that cleavage is happening with hydrolysis and forming adenosine. And so from there, this methyl donator actually goes through a methyl cycle that helps keep regenerating methionine, and also the methyl donator. So when you have that active methyl group that leaves off, remember again, form that S-adenosyl homocysteine. You have hydrolysis to form homocysteine. It can react with activated carbon structure to form that methionine that we kind of seen before. And then it can react with ATP to regenerate SAM, which is once again that S-adenosyl methionine. So, so this homocysteine, it is also used to react with serine to actually end up forming the cysteine. It goes to de dehydrogenation, dehydration, excuse me, to have a condensation type of reaction, and then from there go through hydrolysis to cleave the bond to end up forming your cysteine, and you have this alpha ketobutyrate. And then now, finally, you have the biosynthesis of aromatic amino acids, and how that happens is you have this phosphor phosphor phosphoronol pyruvate do a condensation type of reaction with urethros 4-phosphate. Um, and this is hydrolysis to have this condensation happen. And then from there you have NAD plus. Remember that is oxidation. Um, yeah, it's oxidation and ends up forming this double bond oxygen. And also notate that the phosphoryl group is also released. And then from there, you have the intermediate is dehydrated to end up removing the hydroxyl group to have this key intermediate, the shikimate intermediate, and then from there goes to NADPH, and that is once again reduction, and so that double bind you end up adding a um, having a hydrogen group that is added, and then from there you have an ATP dependent reaction where it is adds a phosphoryl group there, and then it reacts with the phosphoronol pyruvate, and uh, the phosphoryl group also gets released, and it's just the carbon skeletal that is attached to the key intermediate. And then from there, the phosphoryl group is released to have this next key intermediate. And then you have um, some conversion that ends up happening, and then it has two different paths to form the aromatic amino acid. And so you have NAD plus, which does oxidation. Um, so it takes up that hydrogen that's right here, form this intermediate, and then reacts with glutamate to form alpha ketoglutarate. And also the final form of the amino acid, tyrosine. And then another option is, or a path, is dehydration and decarboxylation to form the phenylpyruvate. It, it reacts with the glutamate to form alpha ketoglutarate, and the final form the phenylaniline. And that concludes the second section of the biosynthesis of amino acids.